All right, guys, we have a great episode of Hairy Matters coming up with Dr. Kevin McKelwey. Before we get into the episode, I will mention this is a sponsored episode, sponsored by Antigen.xyz, HairDAO's very own telehealth service. We have 18 different hair loss treatments. Some are patented, some are just hard to find, some interesting combinations you can't get from elsewhere. So if you like hanging out with your friends or you're not friends in our Discord server, if you like these podcasts we put out, or if you just want more hair loss research to keep being funded, you should buy from Antigen.xyz. All profits are used to fund further hair loss research so we can keep developing newer and better treatments until we get to our end goal of turning a slick, bald scalp, making it fully hairy again. So with that said, we'll get into the episode soon, but while you're here, why don't you hit that subscribe button? Why don't you look in the description below? You can see the link to Antigen.xyz. And with that, we'll get into the episode. All right, guys, welcome to Hairy Matters episode 10. We are finally in double digits. Today joining me is our guest, Dr. Kevin McKelwey, one of the world's leading hair researchers in the hair follicle biology and regenerative dermatology. He's currently the Chief Scientific Officer of Replicel Life Sciences, where he's helping to pioneer cell-based therapies for hair loss, tendon repair, and skin rejuvenation. He also serves as a professor at the University of Bradford School of Chemistry and Biosciences, and previously conducted postdoctoral research at both the Jackson Laboratory and University of Marburg, two top institutions of biomedical science. Kevin's work sits at the intersection of academic rigor, biotech innovation, and translational medicine, and we're thrilled to have him here to talk about the future of hair regeneration.
Thank you, Andrew. It's good to be here.
Okay, so Kevin, you've worn multiple hats as a scientist, a founder, a consultant, an academic. How did your journey in hair loss begin?
Actually, by accident. I was when I did my first degree, I wanted to do a PhD. I really wanted to do something on autoimmune disease, and so I interviewed in various labs, and one of them was looking at alopecia areata, and I decided I wanted to go do that and to do some research on that. But of course, as part of that, I ended up having to learn all about hair follicles and hair biology as well. So that was sort of my first introduction, and then I got interested in it, and that became the main focus for me.
Quick question maybe before we go into some more of the hair loss specific focus, but I saw that you worked at the Jackson Laboratory, and obviously that's pretty fascinating because don't they have some like crazy share of like the total preclinical mouse work that's done in the world? Isn't like an overwhelming percentage from Jackson Laboratory?
Well, it's a supplier of mice for a lot of research, particularly in the US, but also elsewhere around the world. So I think they are the largest provider for mouse models of different kinds for all kinds of different diseases and conditions. So yeah, they produce several million mice per year and send them out to a lot of people. And they also do their own research as well. So they have labs there as well.
Did it impact the way you think about mice as a preclinical model at all? I mean, I know we'll talk about the recent review which you published, but yeah, curious your take on mice as a preclinical model.
Yes, well, I mean, of course, I was exposed to pretty much all the kind of different mouse models that you can possibly have for many different conditions, but also for different kinds of hair loss conditions as well. So mostly I was focused on alopecia areata, and we have a mouse model for that, of course. But all the other models for, things like FGF5 mutations or indeed androgen receptor mutations as well, these mouse models are all available through the Jackson Laboratory. And I was exposed to, most of those models at some point for the 3 years that I was there. Got a fairly broad introduction to all these different potential models that could be used. And yeah, I have to say that's probably what really gives me the background knowledge for me to be commenting on different models and different conditions.
I guess just broadly, there's of course the research joke that you could piss on a mouse and it would regrow hair, but do you feel like the mouse model is representative of sort of in vivo activity, or is it predictive of in vivo activity based on what you've seen?
There's no good mouse model for androgenetic alopecia. There are models, but they're not great. There's always different disadvantages with them. And not least is that we don't really have a good mouse model that actually has hair loss, which is androgen induced. So there are a couple of mouse models that you can kind of use. You have to give them androgen supplements, and yeah, you get some kind of changes to the hair growth patterns and that sort of thing. But they don't really lose their hair, or at least not very much. So it's not really the same as what you see with humans, and that's a problem. But they do give some advantages as well. Not least, you can study them for long term, like months, even a year, up to a year. Whereas with in vitro models, you're only looking at culturing hair follicles for 7 days, 10 days tops, that sort of thing. So you can do some things with that, but there are limits to what you can do because you can't go for very long in culture. So it depends on what kind of question you're asking. If you want to look at the immediate changes to hair follicle growth rates when you put a drug on them, then yeah, you can do in vitro studies. But if you want to look at the long-term factors like the hair growth cycle and how it changes over time, how many follicles are induced in anagen or how many follicles go into catagen, then you kind of have to use a mouse model because you can't do it in culture.
And so just following up on sort of, the trade-offs of mice, obviously, in some cases with hair loss, they're not super predictive of how exactly it might regrow hair or not in human. Safe to say that there's probably a lot of those same problems that you could extrapolate out to other conditions that people are testing.
Yeah, I mean, definitely. So also for alopecia areata, which is the model that I usually focus on, it's the same kind of situation. So, any mouse model, it's a mouse, right? So it has the same basic structure in terms of hair follicles and also in terms of internal organs and all that sort of thing. But there are obvious differences. So the hair cycle is different, for example. So you've got to bear that in mind when you're looking at research using mouse models. For pattern hair loss, yeah, the androgens don't really have the same kind of effect on hair follicles. For alopecia areata, same sort of thing, because the inflammation around the hair follicles in mouse models is not the same kind of inflammation in terms of the numbers of cells and where they focus on, compared to what you see with humans and alopecia areata in humans. So there's always these differences, depending on what kind of model that you're using. So you have to be aware of that. You have to interpret the data that you're getting from a mouse model and say, okay, yeah, this is useful, but, and there's always a but. But that's how it is, right? With whatever model you use, there's always going to be a but somewhere along the line, and you just have to deal with it. Preferably use, more than one model to try and get the same kind of answers. And if you're getting consistent results over time with different models, then that's, that boosts your confidence that what you're seeing is real and what it should translate to what you see in patients and humans as well.
And so you mentioned the alopecia areata mouse model. Curious, what is that and how is it different than the androgenetic alopecia mouse model?
So for alopecia areata, there's a mouse model which is it's called the CDH-HEJ mouse model. That's a particular strain of mice that you have, and they spontaneously develop alopecia areata. And if you leave them long enough, if you leave them for a year or more, about 20% of the mice will develop this alopecia spontaneously. This was actually seen at the Jackson Labs in their standards colony that they're producing. And so this was why I came in and I started doing the research on the mouse model at Jackson Labs. But you can also take the cells from the mouse model that develops it spontaneously and grow those cells in culture and then inject them into normal haired mice, littermates, and actually induce or transfer the alopecia areata. So in that way, it's first of all showing that the inflammation is the important part because you can transfer it using immune cells. And it also shows that all the mice potentially are receptive to developing alopecia areata. So that's interesting to know, useful to know. So it suggests that they need some kind of trigger or some kind of activation event to actually develop the alopecia. And then ultimately we can use that model in different ways. We can look before they develop the alopecia and after and during and see different things that are happening. With androgenic alopecia, the models, again, they're just not really there because they don't actually lose any hair and they don't do it spontaneously either. So what you have to do is you generally have to add androgens into the model at some point. And then this later on will have some kind of change or effect on the hair follicles, usually in terms of delaying the onset of anagen or prolonging telogen. But you don't really see miniaturization of the follicles, and you don't see the hair loss, of course.
Okay, so I think that's sort of an interesting pretext and your experience with the mouse models, and I think to now go into to humans, androgenetic alopecia in particular, obviously alopecia areata as a disease, it's classified as a disease, gets a bit more funding. I think we've looked at certain databases that have found for basic research into androgenetic alopecia, the numbers of the estimates are about $5 million per year globally, which is obviously quite low. You've been around for a while. You've seen all the industry dynamics. In your assessment, why do you believe that funding has been so low into androgenetic alopecia, despite sort of this massive community of people who obviously care a lot about it?
I mean, partly it's because, well, certainly on the academic side, anyway, when you're trying to get a grant application from the NIH or, some equivalent government body, you're up against other grant applications for conditions, anything like cancer, skin cancer potentially. So inevitably, the reviewers look at your application about androgenetic alopecia and they compare that to the skin cancer application, and usually the money goes to the skin cancer. So it's very, very difficult to get a grant actually awarded on androgenetic alopecia. And usually, for the most part, when people submit grants to the NIH, they tend not to talk about AGA. They usually talk about stem cells. And they might introduce androgenetic alopecia as something, as a kind of a model for understanding the dynamics of stem cells in the follicles. But the main focus of that grant application is usually stem cells and their survival and the patterns of growth and this sort of thing. So to some extent, you can try and make your grant application seem significant and relevant beyond androgenetic alopecia by focusing on stem cells, which could apply to many other conditions where stem cells are important as well. But it just so happens that you're using this model of AGA to understand those stem cells. And then in terms of on the commercial side, the business side, the amount of funding, it tends to go in waves, and it's usually in tune with the stock market. Like I can remember there was a fair amount of money coming in back in the late 80s, early 90s, usually around on the back of minoxidil. Minoxidil was being promoted quite heavily back then. And then a little later when Propecia came in as well, finasteride. So there was a fair amount of money up until about 2000. And then, of course, the stock market crashed. And all that money disappeared. But previously, up until 2000, a lot of the, even some of the big companies, pharmaceutical companies, they had quite big labs doing research on AGA. Johnson & Johnson, they had a big lab doing hair research. Pfizer was doing hair research. And various other companies as well. L'Oreal had a big group as well. So there was quite a lot of research up until 2000-ish. And then the crash came. And of course, they cut their budgets quite significantly. One of the first things to go usually is the research side. And then later on, it just never came back. It just didn't recover, even though the stock market recovered. So there's not nearly as much research going on the commercial side as there used to be. But there are still some. L'Oreal still has some labs doing hair research, and a couple of the other companies as well. But for the most part, these days it's smaller startup companies that are doing the research now. It's not the big pharma anymore. So the big pharma, they pay attention. They look at what's happening in the field. They follow along, but they don't really get actively involved in doing the research themselves so much anymore. So that's a bit of a shame, but it's just the way it seemed to have gone.
Yeah, so as people get fat and happy, they start worrying about things like their hair loss, and then when everything goes down, they start thinking about maybe things that might be more essential, I guess. So before we go into some of the specifics of your current company things you're working on, more broadly, what do you think currently is most misunderstood about the biology of AGA? Is it the way the hormones might impact the hair follicle? Is it inflammation, immune response? What do you think? Maybe not misunderstood, but maybe the most underappreciated part of AGA in particular?
Yes, I mean, there's still quite a lot of gaps in terms of our understanding of androgenetic alopecia. We still don't really understand how the androgens actually interact with the receptors and trigger the cascade of events inside the cells, internally. That intracellular signaling system that leads to, senescence of the cells, apoptosis, changing in terms of the hair growth cycle, all this sort of thing. We don't really understand that mechanism very well at all. So that's, something which still needs to be looked at, and I don't think there's that many people actually doing the research on that. But then, as you mentioned, inflammation, and oxidation and all this sort of, the peripheral issues, which could be potentially quite important, they haven't really been looked at in any meaningful way. So we still, again, don't really know what the impact is, whether it's relevant to everybody who's got pattern hair loss, or whether it's just something which affects a subset of patients. Certainly, when we look at the biopsies from the scalp of different people, we can see that there's different levels of inflammation. So it's important for some, but it probably isn't important for everybody. But again, nobody's really looked at it. Nobody knows the significance of the inflammation, what it's actually doing, how much of an impact it has compared to the androgens. This just hasn't been looked at.
So, okay, so moving on to your work with Replicel, I think the first interaction we ever had, I saw a tweet of yours saying cup cells are king, or something
Yeah.
of the like.
Well,
So your work with Replicel, I would give an overview, but I think it'd probably just be better if you did, of sort of what the overall goal was with Replicel, maybe what the initial research, where that came from to sort of base this company, and kind of what your goals are presently going forward.
So, well, the research originally started when I was in Germany for a while, doing research in Marburg, and we were, again, using mouse models. So we started from the mice, but we were taking different cell types from what's called the dermal sheath and the dermal papilla as well, and culturing them in different ways, and then implanting those cells into mouse ears to look at what they could do in terms of inducing hair growth. And so it's not surprising that papilla cells would promote hair growth, because that had been published before by people like Colin Yehoda, of course, and later on Claire Higgins as well. So that was fairly obvious. But what we looked at was the dermal sheath cup, which is basically the bottom part of the dermal sheath, which sort of sits around the periphery of the follicle right at the bottom. It's cup-shaped. And we took those cells and cultured them and implanted them. And we found they were equally capable of inducing hair growth. And the kind of patterns of hair growth we were getting looked better to us compared to the dermal papilla cells. So we were getting a more even spread of hair growth across the ears, and it looked more natural. The angle of the hair coming out of the ears was more natural as well, this sort of thing. So that's why we decided we would focus on cup cells rather than papilla cells. That was back in 2002. And there were some delays because I moved to Vancouver in Canada. So not a lot happened for a couple of years. But once I got to Canada, I asked a colleague who was also in Canada in Vancouver, to help set up a company. And that became Replicel later on. And we decided that we would start to do this cup cell cultures and try and develop that to actually treat patients with pattern hair loss.
I feel like you're the first person I've heard who's really championed the cup cell, at least. And so what do you think? What do you think makes them special?
I'm reasonably convinced that somewhere in the cup there are stem cells, dermal stem cells. Not very many, perhaps. Well, yeah, quite likely. But there will be a few, and those are the cells we really want to get at. So when I look at papilla cells, they're very good for hair growth induction, no doubt about that. But they aren't really stem cells, or at least I don't think so anyway. So although they have some, flexible characteristics as well, but they're not true stem cells, they don't have the right kind of marker profile to show that. And also when you look at the natural hair growth cycle, you can see that the cells at the early anagen onset, the cells from the cup actually migrate into the papilla and create the papilla structure. So the cells in the papilla are not actually proliferating themselves, it's actually cells from the cup which proliferate, and then they migrate in, make the papilla, make the follicle structure, induce the hair growth. And at the end, during catagen, you start to see cells migrating out of the papilla back into the cup shape. So it seems like the cup shape is actually the permanent structure, or the more permanent structure, rather than the papilla cells. And then again, going back to mouse models, and this is studies that have been done by other people, not by us, but they have shown that the cells within the cup survive over multiple hair cycles in mice. And they basically feed into the papilla, and then they migrate back out again, and then feed back into the papilla again, and then migrate out again. So they seem to survive over multiple cycles, whereas the papilla cells don't necessarily do that. They're there for one cycle, and then they disappear. They either merge back into the cup, or they may go into apoptosis, or, there's the suggestion that they also actually migrate to the follicles entirely, and they may actually end up in the the dermis, or even in the subcutaneous fat layer.
So the cup cells might have some form of memory that the dermal papilla cells don't have?
Yes, you could look upon it in that way. I mean, if the true stem cells are actually in the cup area somewhere, they should be permanent. They should last for the duration of somebody's life, and for a mouse's life as well. So they should be there somewhere, if that's where they are. Now, it's fair enough. I should say, not everybody believes the stem cells are in the cup region. They could be elsewhere in the dermal sheath. Nobody's actually found them. Nobody's truly pinpointed, these are the stem cells for the dermal sheath. But they must be there somewhere, because the dermal sheath structure itself survives for, forever, for as long as the follicle survives. So there's got to be stem cells somewhere. And I think they're in the cup, but other people would disagree with me.
And have you seen any of these? I mean, have you thought about or looked into any of these like newer technologies like StereoSeq or these sort of like super high resolution sort of processing capabilities that we're starting to develop? Because I know at the beginning of when these things were being developed, it was like the chip size, an entire follicle was like it couldn't even, the resolution like wouldn't even capture like the follicle. And I'm like, now do you think we're getting close to the point where you might be able to use some of those technologies, I guess, to validate the thesis you've had for a while?
Quite likely, yes. I'm not quite sure we're there just yet. But I think people are paying a lot of attention. It's not just Replicel. So yeah, I think it's also partly costs as well. But I think when the cost comes down to a reasonable level, that you can do a number of decent replicates on different follicles from different people, then I think these kind of approaches will be used, and people will be able to identify stem cells and other markers that they might be looking for in the hair follicles, in the dermal sheath, or wherever else. So yeah, I'm sure it's coming soon, probably.
And then in terms of where Replicel currently stands with its sort of like phase 1's potential phase 2 clinical trials for, I think the way you guys have it broken up is sort of slightly modified treatments based on the indication you're going after, whether it's hair regrowth, tendon repair, skin rejuvenation. Maybe if you could just give an overview of sort of where you are with the phase 1's, potential phase 2's, and then I guess any learnings you've had so far, things you think might be helpful to share, things that surprised you as you get into the clinical stages of developing treatments.
Yeah, so there's actually 3 potential different targets or treatments that we're looking at. So one is hair growth using the dermal sheath cup cells. But we were also looking at the dermal sheath cells, which are higher up in the follicle. These are much more structural cell types. They produce a lot of collagen. And so potentially that could be useful for other situations where you need to repair collagen, basically. So in tendon repair, you need regeneration of collagen, and in skin rejuvenation, you're looking for collagen deposition in the skin as well. So we think maybe the dermal sheath cells could be used for those particular treatments. And then the dermal sheath cup cells, because they're hair follicle inductive, could potentially be used for inducing hair growth and increasing and improving the size of follicles and preventing miniaturization and androgenic alopecia. So that's the main distinction that we're making. So far at the moment, we've had some preliminary studies. We used the dermal sheath cells, not really clinical trials, phase 1 clinical trials, but we've done some clinical work with them. But we have done a phase 1 clinical trial for the dermal sheath cup cells for pattern hair loss. And we had 10 men and 9 women injected just in a very small area on their scalps. And using the cells from the hair follicles at the back of the scalp, culturing those for about 6 weeks, more or less, and then taking those cells and injecting them into just a 2 centimeter diameter area in the areas where they're losing their hair. And they're looking to see what kind of hair growth they had. And overall, about this data has already been published and presented out, publicly. But what we saw was that there was some very good responders. There was 5, 6, 7 good responders. The top two actually had a 20% increase in their hair growth density, which for a single injection I thought was very good. But then, of course, there were others who did not get that kind of level of response, and there were some who did not respond at all. So there was a variation. There was a range. So the objective for Replicel is to improve the quality and the consistency. So 20% injection with just a single injection is great. And again, we have some data from mice, but it shows that you can get cumulative deposition of cells, or if you give them multiple injections over time with some space, time between the injections, you can gradually build up the cells rather than giving them a massive injection. So if you give them a massive injection, just one injection, you actually get a response, but you don't get the same level of response as building it up gradually. So going forwards, our objective will be to, when we go into phase 2 trials, will be to do multiple injections and just build up that number of cells. And we hope that we'll get a better result as part of that. But then we also have to look at the quality of the cells. We need to get to a threshold level, at the very least, of quality for each individual every time. And that's something that we're working on in the lab at the moment. And there's various ways to do that in terms of ingredients, which you add into the media, the ways you inject the cells, and different processes that you can use to actually control the rate of growth of the cells and all this sort of thing. So there's various things which are going on at the moment to actually improve the quality of those cells.
I guess, what are the main sort of phenotypic differences that if anything, that you've noticed so far in terms of dermal sheath cup cells that perform well or those that don't, and whether that's, how you are inducing, or refining or inducing the cells to change to your, desired effect, or whether that's just, some patients have particularly strong and vibrant dermal sheath cup cells.
Well, it's true that some people do have strong and vibrant dermal sheath cup cells and dermal papilla cells as well. So you do see a variation in the cell types and the pattern, in terms of gene expression. You do see differences. Even in the fresh cells, before culture, when you look at the papilla cells and the cup cells and you do some measurements, you will see variations between individuals.
What does a healthy cell look like versus not?
Well, so we have data which, we don't make public, but one of the things that we do look at, which we have made public before, is simply to look at alkaline phosphatase expression. That's no great surprise to anybody. There's lots of data out there about alkaline phosphatase in papilla cells, and you also see it in cup cells as well, albeit at a lower level. So we look for the quality of the expression of the alkaline phosphatase, at the level, first of all, the distribution, the number of cells that are producing it, and then changes in that over time in culture. So that's one of the things. But we have a lot of other things that we do as well to look at the quality control issues, but we haven't made that public at the moment.
I think your results were the best in Norwood 3 and 4 patients, and so I was curious just for your take, because I think this is pretty common across all hair loss treatments. It's like, it's even with finasteride, right? It's like, take it before you have lost it all, because it's better at maintaining than sort of reversing hair loss. So what do you think it is that does limit the effects in the later stages, somebody who's suffering from hair loss?
Well, of course, pattern hair loss is progressive, and, the problems are cumulative. So in the early stages, most of the issues are changes to the hair growth cycle, but not so much in terms of the size or, activity of the follicles themselves. So you've still got the right numbers of cells there, pretty much at the early stages. But then as time goes on, of course, you start to lose cells from the papilla, from the cup, from elsewhere, partly through senescence, partly through apoptosis, and so you get the miniaturization of the follicles. So then you're not only trying to counteract the, improve the hair growth cycle, now you're also trying to regenerate the follicle and get it back to a larger size again, get it back to full size again. And then, of course, as time goes on, there's more miniaturization, and you get to the point where you have vellus hair follicles. So then you've got a much, much bigger challenge, because now you're trying to regenerate that vellus follicle into a full-size terminal follicle again. So there's a lot more to do. There's a lot more for a cell treatment to overcome, first of all, or even for a drug to overcome, versus, if you start early, when you've had your patent hair loss maybe just 2 or 3 years, and there's a little bit of damage, but it's not too bad. The follicles can bounce back again if they get a reasonable kick with a drug or with a cell therapy treatment to get them back to full regrowth again. Versus, later on, when you've had it for 10 years, 15, 20 years, then, there's a much bigger challenge to regenerate that follicle, get it back to full size again, get it back to full growth again.
And so you mentioned there was some tweaking and refining that needed to probably happen before you think that you guys would be ready for sort of a phase 2 trial. So I guess I'm curious, what have been kind of the biggest bottlenecks in getting to that phase 2? Was it just sort of you need to tweak and refine more until the efficacy kind of is more exciting for people to fund a phase 2? I guess tangentially, is it funding? Are there regulatory hurdles that cause you issues? I mean, I'm sure it's a combination of the three of them in some shape, form, or the other, but I'm curious to hear a bit about that.
Yeah, I mean, it's a combination of all of those things. So that the regulators have been quite a significant issue, because at the moment, we're talking to, recently anyway, we were talking to the European regulators because we were planning to do our phase 2 trial there. That may not happen. We may move to another jurisdiction, may come to the US even. So now we've got to talk to different regulators. Of course, when we were collaborating with the Japanese company, we were talking to the Japanese regulators for a while. And each regulator or regulatory system has its own different requirements as well. So you've got to try and meet the requirements of all the different regulators and what they expect. So it can be quite challenging in that way, that there's lots of things you need to do before they're willing to accept your clinical trial proposal and give the green light for it to go ahead. But it's not impossible. It's not insurmountable. And then the other issue, of course, is funding, inevitably, because clinical trials are expensive. It'll be multiples of millions. It was several million just to do the phase 1 trial. Phase 2 will be much bigger, so it's going to cost more. So, of course, you've got to get funding for that. And then in terms of the quality control, we're fairly confident that we know what we want to do, what we need to do. We will have to do some further work in the lab to validate the new protocol, the improved protocol, before we actually do the trial, because that data has to be presented to the regulators. They want to see that and show that you've actually made improvements and that you're getting consistency and that you've got your quality control and that you're also making sure that the cells are safe and, there's nothing strange going on in the cultures that they might be worried about. All this data has to be presented to the regulators to get their approval.
And so, okay, so it's not just a lot of sort of bureaucratic paperwork, but then also just a lot of data gathering that they just kind of keep firing more and more questions back at you. Like, has there been any X, Y, and Z expression in the cells that could potentially be dangerous?
You get that quite a bit, yes. We had that for the phase 1 clinical trial, and we will expect to see that for the phase 2 trial. So now they're expecting that they'll want to see the phase 1 clinical data, of course. They'll want to see the improvements. They'll want to see much more in terms of quality control going to phase 2, and also safety data as well. But we have that from the phase 1 trial. So it's, as you say, it's just data collection. We've got to collect a lot of data, put it into a format which is, the appropriate format for the regulators. And then they go through it, they digest it, they probably come back with questions, they always do, which you've got to answer. And sometimes you may need to do more experiments as well to, get more data to answer those questions. But eventually, hopefully, you get to the point where they will agree to approve the clinical trial.
And do you notice some jurisdictions perhaps being like more scientifically literate and then maybe asking more relevant questions and less of kind of the busy work questions that kind of keep you constantly working to get to the actual trials?
Yes, I mean, you see some of that. I have to say the regulators certainly in the US, Europe, and Japan, They're not particularly, they're not that dissimilar, right? So they're all going to ask for the same basic data and information. They're all focused on safety. They're all focused on, making sure that you've got a safe treatment that is going to be effective to some degree in everybody. So those requirements are consistent across the board. It doesn't matter where you go. It's just a challenge in terms of the approach can be a little bit different. The kind of questions that come back from the initial data might be a little bit different. But ultimately, they're all pointing in the same direction, which is, safety and making sure that this clinical trial is not going to be a concern for, side effects and issues with for the patients.
This is just a community question that people seem to be quite curious about. But have you guys at Replicel been able to confirm integration or like homing, if you will, of the injected dermal sheath cup cells into the hair follicle niche?
So we have mouse model data. And then when Shiseido were focusing on this, they also did some work where they'd used human tissue, but outside, away from patients. So this was, in culture and that. And they were injecting labeled cells, cup cells, and showing that the cells actually migrated and integrated into the follicles. And they did publish that a few years ago, although it was kind of buried in a bigger paper. So they did show that the cells do migrate into the follicles. There's a lot of cells that stay around the periphery as well, so they don't all migrate in, but there will be some, and some of them do get to the dermal sheath cup and into the papilla as well.
And I mean, you kind of answered this earlier as well, but this is the second community question, so we like to make sure we get them in. But in terms of Replicel being different than like another company like Adirans, I don't know if you've seen them much, but is it really just the cell type that's the main difference? Sort of, they're using dermal papilla cells and you're using dermal sheath cup cells?
I mean, I don't know the full details, of course, for the other companies, but yeah, I mean, the main difference would be that we're using the cup cells and they're using papilla cells, as I understand it. Although if it's Adirans research, they as time went on, they changed their sort of focus. So originally they started out on papilla cells exclusively. But towards the end, they were actually taking biopsies from patients and then making an extract from that and culturing a range of different cells and then using those. So they weren't actually at the end using papilla cells exclusively. So it did change over time.
Now I think it'd be cool to sort of go to the paper you recently published, and sort of I really enjoyed reading it. It was like a great overview of kind of, I mean, I felt like if you were somebody who was getting interested in hair loss research or something like that, or you had been dedicating a lot of time to it, it was like an excellent map of sort of the current state of hair loss research, and like the best ways to kind of pursue whatever answers you're looking for with the models that are available. So I guess, fair encapsulation of what you said earlier would be to say that of the models that exist, it's sort of, different tools for whatever your goal is. Like obviously if you're super interested in prolonging antigen, for example, like in the short term, maybe the human hair follicle organ culture Philpott model is the best. But if you want to study over time, you got to use a mouse model.
Basically, yes. So, I think very well, Andrew, I think from the studies that you're doing right now with the hair follicles. So you can certainly put the follicles into culture. You can add your, whatever factor it is, your drug, whatever it might be, into the culture media. You can see what effect that has on the hair follicles. And if it stimulates faster growth, maybe, or if it stimulates growth for a longer period of time in culture, that's very, useful and significant to know. Still doesn't give you the whole story, of course, because you don't know about the hair growth cycle, and you also don't necessarily know about what it might do to follicles that are miniaturizing or even vellus follicles. So, there are limitations there. So if you want to look at the hair growth cycle, then you have to go to a mouse model. But then there's the limitation that they're not a good model for pattern hair loss. All right, so you can get some information from different models. You have to sort of add it all together to get your full overview, your full picture. Which model you use depends on what question you have.
Claire in the last episode did touch on the fact that maybe it's because of the model limitations, but the the cycling is relatively less well understood than the how to prolong the anagen phase.
This is the biggest issue. We don't have a good model for pattern hair loss and looking at the hair growth cycling changes. The mouse model is no good. In vitro models just don't cut it either because they're just too short term. So what can you do? We're kind of stuck, right? The model for pattern hair loss, they just haven't been developed to the point where you can use them reliably and get a good result and be confident in that result. And models for other forms of hair loss, even for alopecia areata, are much more developed. They're better developed. We understand them more, and we know that they reflect the human situation much better. So there's more progress because we can take that data that we get from the mouse model, use it with more confidence, and also the in vitro models as well. We can use it and feel confident about that data.
And then even on top of that, another thing which I think you mentioned in your paper is that when we do use hair follicles, we typically are using occipital hair follicles, which don't really miniaturize in the first place. And so do you think that's just a function of hair transplants and how they work, and people are taking hair from the back of the scalp, and so it's just easier to get those? Is it really just that simple?
In large part, yes. So in terms of what's available, if you want to look at human tissues, hair follicles from the back of the scalp are, relatively speaking, available because of the hair transplant clinics. So I think that's pretty much where all the hair research groups get their hair follicles from. They have some link to a transplant clinic nearby, as we do, as Replicel does. So that's our supply. So then we have the limitation that, these are not very androgen responsive follicles. So again, we have to interpret the data that we're getting. If we're looking at pattern hair loss and trying to interpret the results we're getting in vitro to the actual situation in humans. For a few groups, occasionally they can get androgen responsive follicles because there are facelifts which are done, or brow lifts, and they actually do a cut, an incision up here behind the hairline. So sometimes you can get androgen responsive follicles, but it's it's more challenging to get those tissue samples.
I think you also touched a little bit on iPSC derived follicle models. I actually know less about those, so curious just to get your overview and take if you think they are a meaningful step forward in terms of being representative of in vivo activity, and if so, I guess why or why not?
I'm not an expert either. We've sort of dabbled a little bit in the lab, but we're not experts in any way. I do think that they've got potential as a much better model compared to what we have at the moment, because you can control the kind of cells that you're using, and you can also culture them for a fairly long period of time relative to what we're using at the moment. So, potentially we could be able to start looking at certainly hair growth induction and the early anagen phases, and maybe we're lucky and we start to look at changes in the cycling using iPSC models as well. But I think the big advantage, at least in my mind anyway, is that we could take androgen responsive cells or make the cells in the iPSC models androgen responsive. And so maybe we get a much more accurate reflection of pattern hair loss. It would have to be tested. This model isn't developed as such. But if you could use androgen responsive papilla cells in your iPSC model, and then you add in your androgens into the culture media, and you see changes in the follicles, miniaturization ideally, then that would be a very, potentially a very good model to be using for screening drugs or for whatever other studies that need to be done. So we're not there yet, not by any means. There's a lot of work that would have to be done, but I think it has a lot of potential.
Yeah, okay, artificially increasing the supply of frontal follicles that we can test on. Look, hopefully it's almost like you're getting close to hair cloning at that point, right? Where you could theoretically, just generate a bunch of new follicles and transplant those into somebody.
There are companies that were working on it until quite recently, and So I don't know what the details were, whether they were making progress, but certainly that's in people's minds. Yeah.
The existing field and the progress that's being made. This is sort of a two-part question. The first part of the question is, what is your take on uh the potential for newer small molecules like Wnt activators, thyroid agonists, prostaglandin analogs, and then also just really like, what is Kevin McKelwey's hair loss stack?
Yeah, I mean, I think the future looks relatively bright at the moment. So, I mean, there are a number of clinical trials being done, whether it's what's it, Claxturone, or whether it's the Wnt activators. There's various things that are being developed and that are in progress. And I would expect to see some reasonable results out of quite a few of these different trials as well. Yeah, why not use Wnt factors to promote hair growth? That seems entirely reasonable. I think it comes back to what we were discussing earlier, though. What stage of alopecia do you have? So if you're in the early stages of pattern hair loss, I would expect to see these factors having much more effect. So the Wnt factors they've got more to act on. And the follicles are not that particularly damaged. They're damaged but not too far gone. So the Wnt activators should be able to regenerate the follicles quite well. But then you start to get to, you've had 10 years or more of pattern hair loss, your follicles are relatively miniaturized, they've been damaged for a long period of time, you've probably lost some of the stem cells in there. Then the Wnt factors, I mean, they're in themselves just as effective, but they don't have as much to act on. You don't have as many cells there for them to act on. At a very basic, simple level of looking at it. So there's less that they can do, right? They've got less to work on. So again, I think it's going to be much as we see with minoxidil, finasteride. If you're in the early stages, you'll probably get a good response. If you've had it for a long period of time, then I think there's going to be relatively less response. And then that's why Replicel is looking at cell therapy of some kind, because then I think, at the point where you've had it for 15, 20 years, we really need to regenerate that follicle. Maybe that could be done with drugs, possibly, but we think it's more likely to be done with cells.
And so, if you were gonna take like a hair loss treatment or a stack of treatments, what would be your go-to? Obviously, factoring in your own tolerance for side effects, your desire for massive regrowth.
Well, I mean, I'm still waiting. I will get the Replicel treatment when it's ready. But as you can see, it hasn't been done as yet. I have used finasteride. I did use it for a while. It was actually in a trial for one of the studies a long time ago. But I actually didn't get much result. So they measured my hair density at the start and at the end after a year, and I had no change whatsoever. But it didn't have any side effects either. So, I didn't have any problems.
Yeah. It was oral finasteride?
It was oral finasteride, yeah, 1 milligram per day. So yeah, for me it was no problem, but I didn't get much hair growth. And this was actually 15 years ago now.
Did you still have like a relatively full head of hair then? And it just
I had a lot more hair back then, yes.
So it wasn't the case of like the follicles that are already fully miniaturized, and it was just.
I don't think so. I certainly wasn't this far gone anyway. Yeah, I also tried minoxidil very briefly, but I found it was so irritating to my scalp, I stopped after a week. So yeah, maybe it's got potential, but the reaction that I had was just too much. So I didn't pursue that any further.
Yeah, when I first started taking finasteride, I remember just being like really terrified by the initial shed. Like it was, it got better, but at first I was like, oh my God, all of my hair is falling out.
But that's probably something that we should expect anyway, because follicles, you can't change the size of a follicle that much, when it's in midstream, as it were, when it continues to grow the hair. It seems like the follicles have to shed their old hair, go through the full cycle, go back into an anagen phase, and then get hopefully bigger again in that early anagen phase, and then grow a new hair, and then you get a better result. So this just seems to be the case. It was the same with minoxidil, same with finasteride, and some of these other treatments that have been, suggested as well. It seems to be essentially the same issue every time. So in terms of changing the follicle size, probably this is something that has to be expected.
Touching on, I guess, the last two pieces, because you kind of have been and are everywhere throughout hair loss. You also have McKelwey Consulting and keratin.com, which is an incredible domain name. yeah, curious kind of what your goals are with each of those, what what you're working on, and yeah, I think people would love to just know.
Yeah, okay. I mean, keratin.com, I've had that since what, 1998, 2000, something like that. Anyway, um, yeah, I mean, for me, I started it out just as something that was for personal interest. I was trying to get interested in hair and beyond alopecia areata, and I was just reading up about all these different things. And there wasn't much on the internet at the time, because this was, again, 15 years ago and more. So I felt that why not? Why not write some of these up as little statements about different hair follicle treatments and different kinds of hair loss and that sort of thing. And for a while, keratin.com was probably certainly the biggest place anyway for looking at or finding out about other forms of hair loss other than androgenic alopecia. And it was like that for a couple of years, but then that kind of disappeared. So now I deleted it in the end because it was getting too old and I wasn't updating it. And now I've decided to start again, but now it's just sort of a blog, really. Just taking different aspects and writing it up and putting a few references in at the bottom, which I think are relevant. And, if people want to read it, that's great. And if they don't, that's okay. Yeah.
I feel like if you ever wanted a summer home or something like that, that's a pretty valuable domain name that I'm sure somebody would be willing to shell out for.
Yeah, I've had some offers and I've said no.
That's amazing.
I like it too much.
Yeah, we all appreciate your your dedication.
Yeah. What was the other question? I've forgotten now.
Just what you're doing with McKelvey Consulting.
Yeah, so I do a little bit of consulting work for companies. At the moment, not too much because I'm actually working mostly for Replicel. But there are two companies. There's one in France, one in the UK. And I'm just helping them out with one of them's got a clinical trial that they want a bit of advice on. I give them some information about that. Another is doing some lab research work, and I help them with that. It's a sideline, right? It's not the main thing that I do, but it's interesting, and to see how other companies approach the situation. That's quite interesting for me.
Okay, so the closing final question, which I think everybody's always waiting with bated breath to hear what the experts think. But do you believe that turning a slick, bald scalp and like getting it to be fully hairy again by whatever means is possible? And if so, on what time horizon?
I watched the interview you had with Claire Higgins, and I noted that you talked about Dr. Suji. I might not be saying his name right, but if you're talking about an extensively bald scalp, and I'm getting there, as you can see, I think his approach is probably the right kind of thing. I think there's a lot of challenges, certainly, that he's going to face, or he is facing right now. But the principle of making protophorical structures in culture in some shape or form, and then transplanting those is probably the best way to go right now. In terms of extensive pattern hair loss. In principle, I think it should work. I think it should work very well. I think the bigger challenges are actually not the actual getting it to work. I think the challenges are going to be with the regulatory system and with the costs involved, because there will be quite high costs.
No, I think we're going to have Dr. Suji on in the coming week, so we'll grill him about that on future episodes. Well, Kevin, thank you so much for taking the time. I feel like this is I really, really enjoyed this episode. And I know everybody is cheering you on with all your work with keratin.com, with Replicel, of course. Yeah, I guess what's the best way for people to reach out to you or get in contact if they're interested in doing some work with you?
Sure. Well, I mean, you can always contact me through the website, keratin.com. It's got a little page there where you can fill in the details. Or indeed, you can contact me through Twitter. Well, X as it is now.
That's what I did. Yeah, that's what I did. Okay, well, great. Thank you so much for taking the time. And yep, that's been another great episode of Harry Matters.
Yeah, it's been good. Thank you.